have Dr. Amjad Hussain, who will be talking to you. And yeah, Dr. Amjad, the floor is yours.
Right. Um, Thanks IGM team for inviting me as a speaker. So I'll be talking about tumor microenvironment, its genomic information, and the next generation of therapies. This talk would not really fit into the modeling aspect a lot, but I would try to cover some of the modeling part as well, and how the modeling of genomic data can help in sort of treating real tumor patients better, and what are the outcomes and how that can improve the life of a cancer patients. So I'll also cover some of the clinical trial data which was done on real cancer patients, and this is something which is important, that basically talk about and cover something which is beyond in vitro and animal experiments. So that is really something translational which I would like to share with you today. Before I get started, I would like to disclose that part of the data which I'll be presenting was also done in collaboration with ImClone system, which was later on taken over by Lilly. So that part belongs to Lilly, because I was like the research was funded by Lilly. And also a technology which I will talk about was also developed in collaboration with Amgen in United States. And the modeling part is also I will cover from Cellworks. So if you see new cancer cases at present, and like maybe 10 years or 9 years from now, it's constantly increasing, and there is no doubt about it. And particularly if you see the numbers in context with India, then there is more mortalities now if you compare in 1990s and 2000. So definitely, like in 2020, the mortalities are more in India as well, although the healthcare is better. We do have better coverage of insurance, but still. And among those cancer cases, you could also see that every year there are almost roughly a little less than 2 million cases that are adding across the world. And uh, the fraction of it is actually in India. And these are the total cases in India. And 0.8 million is almost the death. And among all those cancer types, the oral and lung cancer are the most common in men, and in women, the breast, oral, and cervical cancer are the most common. So these are the solid tumor types which are more prominent in the population. If you look at the basics of cancer, the cancer, everyone knows that it starts primarily as a lump of the cell, which is a result of uncontrolled cell division. And at that point, any cancer is called primary tumor. But let me tell you, I'm sure you're aware about, but still, just for the sake of highlighting the importance of this migration of cells, that when primary tumor develops, it is generally not harmful, because it is just a group of cells which is sitting in one organ. But the real problem starts when the proliferation is more and more, and these cells start shedding off from the main primary tumor and start moving to different parts of the body, to the lung or brain or kidney or other places. And that process is called metastasis. And that's where we start talking about that, okay, tumor has spread it. So, and then once, for example, if these colon cells, if they move to lung and get metastasized, then they start affecting the function of lungs, and that's how it leads to the mortality and death.
Dr. Amjad, if I could, your slides aren't changing. You're still on the first slide in the.
Oh, really?
Yeah.
Okay. How about now?
No, it's still on the first slide.
Oh, so I haven't been constantly moving. Now? No?
I mean, you're on the first slide right now.
Oh. I'm on the fifth slide, so I don't know what what am I doing differently?
Yeah, now you're on the fifth slide.
Okay. And is it moving now?
It's still on the fifth slide. Yeah, now it's moving. Yeah.
Okay. Cool. Okay, so yeah, so I was giving some of the basics about the metastasis. So the real problem happen when primary cells start moving, and that causes metastasis, and that leads to the organ failure and different sort of malfunctions of other organs, and that leads to death of the patient. So, in order to metastasize itself, the tumor needs to grow beyond its minimal size. And what happened that in order to grow beyond minimal size, it requires a lot of nutrient and therefore also blood supply, because nutrient and oxygen that is provided by the blood supply, and that is why tumor cells, they start secreting some of the factors which start attracting new blood vessel formation and also the blood vessels towards tumor. And for example, VEGF is one of those factors, which is called vascular endothelial growth factor. And then this new blood vessel formation is called angiogenesis. So angiogenesis is not uncommon. It is not only restricted to tumor, because when a baby is born or when a zygote develop into the embryo and then later on the blood vascular system is formed, there is angiogenesis, and there is a lot of angiogenesis in newly born baby as well. But this is something which happen in abnormal and pathological conditions, and these blood vessels often they are not very structured as well. So they are a little different structurally, and I'm not going into the detail. But the bottom line is that if there is more angiogenesis, there is more chances of tumor growth, and it can grow beyond its minimal size. So in a way, angiogenesis is directly related to tumor size. Now the question is how angiogenesis also helps in metastasis. It does, because when these vessels are formed, these vessels do not have very tight vessel wall, because these are newly formed vessels, and pericytes and endothelial cells, they are still sort of making the layers. So these cells have a little bit of leaky wall, and that gives the opportunity for tumor cells to enter into these blood vessels and ultimately becoming the part of the circulation, and that's how they move to other organs, where they also sort of get released out from the vessels through diapedesis, and ultimately they find their new home after metastasis. So in a way, a tumor angiogenesis acts as a double-edged sword for the patient, because at one end it is helping increasing the tumor size, another end it is also increasing chances of metastasis through newly formed blood vessels. So these are the two gentlemen and very prominent scientists from United States. One worked at Harvard, who also happened to be my postdoc mentor, and another one at Genentech. They discovered VEGF long back, and it was named because it has also caused leakage of vessels, and it is also named as vascular permeability factor by Hal Dvorak, while Napoleon Ferrara, who was heading the division at Genentech, he named it VEGF. Now he's, I think, at UCSD. And later on, of course, if you don't know, Genentech is one of the most innovative company of biotech in United States, which was quite a few years ago was acquired by Roche for $49 billion. So the idea is that if we cut the angiogenesis inducing factors, and if we target them to cut the blood supply, can we control the tumor? Instead of targeting tumor cells, can we target the endothelial cells or cells which are participating in vascular angiogenesis? And there are a couple of drugs which are already been approved. VEGF inhibitor Avastin is one of those which is used for many uh, solid tumors, but first it was approved by treatment of colorectal cancer. And then there are other VEGF pathway inhibitors as well, like sunitinib and sorafenib, which doesn't act directly on VEGF, but VEGF also leads to sort of activation of pro-proliferative pathways and pro-survival pathways, so they act on it. Also, there are traps which are kind of have a fusion with protein, which consist of some of the domains of receptors like VEGF receptors, where VEGF binds, and then they can also neutralize VEGF if when they're used. So these are some of the examples of drugs which are approved by FDA, and some of them are also into trials. And recently, some of the VEGF inhibitors are also being repurposed. They're still in trial for COVID as well. But the question is that these targeted therapy, they don't always work. And Avastin is one of the example which often fails. And I met this lady in Boston who lost her husband because, due to colon cancer. And the reason was because in late stages, Avastin stopped functioning. It becomes, non-efficacious in later stage. So the question is, why do these patients don't respond to therapies? Like once they're responding, and then after a while they stop responding, and that's where the modeling and signaling pathways also become important. But before even I move into it, let me give you the idea that tumor angiogenesis is not only governed by VEGF or VEGF receptor 2. This is just one end where it initiates, but there are multiple other pathways, there are multiple other proteins which also sort of promote those proliferative and pro-survival pathways, and still, using VEGF inhibitors are not enough. And a lot of times, especially in late stage tumors, these vessels, they stop expressing VEGF receptors. So they become VEGF independent. So in late stage tumors, even if you use VEGF inhibitor, so generally it doesn't respond. And the reason, because the signaling is also very complex. It is not only one form of VEGF, but there are other forms of VEGF which sort of signals to the cell through different receptor other than VEGF receptor 2. So for example, VEGF C, it binds to VEGF receptor 3 as well. And there is also pro-proliferative pathway. So if proliferation is sort of happening due to the dominance of VEGFR3, then VEGFR2 inhibitors probably will not work. So the outline of my presentation is basically why this resistance comes up and what is the signaling behind it. And then can the sequencing of tumor help us in designing, or at least selecting therapies smartly, so that we can have a better success ratio instead of having failures and waiting whether the patient will respond or not. So what I'm going to present in one or two slides, that how we sort of started in vitro on cultured endothelial cells, compared signaling side by side using different forms of VEGF like A, C, and one of the mutant of VEGF C, which binds to one receptor like receptor 3 but doesn't bind to 2, and also VEGF E, which only binds to receptor 2. So we used a mouse tumor model, mouse angiogenesis model, where you can inject VEGF A, which is expressed in an adenovirus system, or VEGF C, or another form of VEGF, and then you could see that it can cause angiogenesis and formation of blood vessels on day 5 if you use higher plaque forming units of adenovirus expressing VEGF-A. And this is just to show the mRNA and ELISA data that, okay, expression is happening at different time points. So if you see the real experiment and compare VEGF-A and VEGF-C, you could see more blood vessels which are formed, induced by VEGF-A as compared to VEGF-C. The response is mild. And also with VEGF-E, the response is like really, really robust. So it seems that majority of the angiogenesis is happening through VEGF receptor 2, because VEGF-A and VEGF-E, they primarily bind to VEGF receptor 2. When we look at the cross sections of these blood vessels, you could also see that VEGF-A and VEGF-E, they can form large and different type of vessels as compared to VEGF-C. So it seems that, if we treat all blood vessels or every sort of tumor, every type of tumor angiogenesis using one inhibitor, that is not the right approach. Even their expression profiles are different when they are induced by different cytokines. So with that, the Lilly comes into the play, and then what they have done, they have designed inhibitors for different receptors. So there's an inhibitor which is called MF1 that inhibits VEGF receptor 2, DC101 for VEGF receptor, VEGF receptor 1, receptor 2, and receptor 3. And if you see that when you use receptor 2 inhibitor, then angiogenesis is gone. But if you're using other form of inhibitor, for example, receptor 3 inhibitor, it is not working over here, neither the receptor 1 inhibitor. So seems that the specificity is important, and yeah, we have to choose the inhibitor accordingly. Now coming to the mechanism that how it is happening. So basically how VEGF stimulate proliferation of endothelial cell, that VEGF actually activates PI3 kinase that leads to, through multiple pathways, phosphorylation of AKT at two different amino acid site, which is serine 473 and threonine 308. And then activated AKT leads to activation of mTOR through phosphorylation, and mTOR itself is a kinase that leads to phosphorylation of another protein which is called 4EBP, which when it is non-phosphorylated is kind of inhibitor of initiation factor, but when it gets phosphorylated, then it gets detached from the initiation factor. Initiation factor becomes free, that leads to initiation of translational machinery through sort of assembly of two subunits of ribosome, as well as initially the subunit of a smaller unit of ribosome to mRNA. So the point is that ultimately VEGF leads to induction of protein synthesis, also that also leads to turning on of many pro-proliferative pathways. And this slide just to showcase the in vitro impact of using inhibitor of VEGF receptor 2. If you see it here, when there was no receptor was used, the AKT phosphorylation is higher when induced by VEGF A or C. But when these inhibitors are used, the phosphorylation is almost gone. So in vitro data also recapitulate what we see in animals. So the take home is that A or E form of VEGF, they cause much stronger angiogenic response as compared to VEGF-C, means it is coming mainly through VEGF receptor 2 and not through the receptor 3. Now, why am I emphasizing these receptor thing and so much emphasis is given on the animal studies and all? Because it is important that you know what inhibitor you are moving forward when you are working with industry, or if you think about the industrial perspective, then they would like to move on with the clinical trials only with inhibitors which are potential in animal studies. So that is one of the reason. And just to give you the perspective, since many of your presentations and most of your teams, they are leading to some of the product which will be used by customer or will be launched in the market. And if it is a drug, or if it is even anything which is mostly drug or a concept which ultimately will be consumed by human, then you also need to conduct, or someone need to conduct a clinical trial. So just to give you the perspective that most of the research that you do in your academic labs, that is restricted to this sort of step, which is presented here as T0, and that is mostly the preclinical, which is in vitro studies using your cell culture model, and then animal studies. And then once you have a potential animal data, then the molecule, which is important, whether it's an antibody or it's a small molecule, that move to the phase 1 clinical trial. And I'm sure you hear these terms a lot these days in relation to COVID vaccine or new drug discovery or repurposing. But This is really important to understand because you may have a very good working molecule in the lab in vitro and even in animals, but it may not be safe. And the phase one takes care of the safety issues, that okay, the drug is not causing any harm or any organ damage. And then phase two is efficacy trial, and phase three is also efficacy but the randomized trial. So I'm not going into the detail of the clinical trials, but I'm just giving you the idea and just to build on this perspective that where you are when you are doing your research in mostly in the academic labs. And that is why even to move forward, even to transfer the technology to a company, it make more sense if you also have animal data or animal studies. And that's what these big companies do, that either they have in vitro data, in vitro studies done in their own lab, or they collaborate with different academic institutes, and then the animal data or in vitro data that can be transferred, and then if it has a potential after validation, they can move for the clinical trials. So there are these are multi-billion-dollar corporations, and that's how they develop drug and launched in the market. Um, so coming back to the second part of my talk, which I mentioned in the introduction, that how the sequencing information, either NGS or like having some sort of sequencing information, can help in making a decision for therapy selection for cancer patients. And that is also a concept behind upcoming area of personalized medicine or personalized oncology. And this is really important to realize, because a lot of time there are standard procedures and there are standard of care drugs. So for example, if a patient has a colon cancer, and it is quite well known that EGFR in colon cancer is upregulated, so a lot of time there's a standard treatment protocol that okay, patient will be given EGFR inhibitor. But there are cases where patient have colon cancer and they have EGFR knockout, they don't have EGFR gene. So in that case, EGFR gene will not work. Sorry, EGFR inhibitor will not work because they have a deletion of gene. So that is why it becomes very important to have a personalized information of the patient or patient tumor, and then at least, if not the complete information, but at least the key genes people should see, or clinicians should see, whether these are expressed or they are deleted, or at least deleted or present in wild type form, or if there is a mutation in that. So this precision medicine initiative is now, it is actually in clinic already. It's being practiced, especially in Western countries, and there are more than, I would say, 50 companies already operating. If you only talk about California, Silicon Valley, as well as Boston area, so there are more than 50 companies operating in personalized medicine, and I think 7 or 8 of them are, more than $1 billion companies. So the idea is that you have the sequencing information, and then using that sequencing, if you could model those using simulation technology, that okay, what aberration or what deletion means what, and then ultimately, if that data can be interpreted, and then whatever you are predicting, if there is a validation using a case study, then ultimately you can say that okay, you have developed a model and it's working. So that is the whole idea. And then when I'm talking about case studies, so it could be also like the blinded study on retrospective data which is already published, and see whether your prediction is matching. And then of course one has to go prospective if you really want to add value in treatment of patient. So the very famous and one of the very big trial, which was called Shiva trial in personalized space, was done in oncology. And let me tell you, it was a big failure. And the reason being, although they have done personalized trial, they have used personalized approach, but what they did, they selected patient and they see that okay, in this patient PI3 kinase gene is overexpressed or upregulated, or maybe they have high copy number, so they given them PI3K inhibitor. In some of them, let's say MEK has high copy number, so they have given MEK inhibitor, so something like that. Or in case of breast cancer, if there are these hormonal receptors, ER, PR, so then they have given those inhibitors. So accordingly, they make the group. But then they did not consider multiple changes or multiple mutations. So for example, if PI3 kinase is there, but if AKT is deleted, then what is the use? Because AKT is downstream. And if it is deleted, then PI3 kinase will not be able to exert its impact. So that is why it was one of the reasons why this trial was a failure. And if you could see that with a personalized approach and without personalized approach, there was only a difference of, like, 2.3 months of extra survival versus 2 months of extra survival, which could be like an error. So it doesn't mean much actually. So, but the sequence-driven personalization, it does have some of the gaps also, that they talk about only one or two genes, one or two mutations, but there is no information, no FDA guidelines on, let's say, high copy number of genes. For example, one case could be that, okay, one of the pro-oncogene is mutated and it has gain of function mutation, so it is more active. Another approach could be that the gene is not mutated, but it has high copy number and it has high expression. So the protein is being made in more quantity, and then it is sort of, having more impact. So basically, high copy number may also have a similar impact of what a gain of function mutation is doing. But then there is NCCN guideline of treatments on mutation, but there is no guidelines on copy number. So these are some of the factors which should also be considered when we model or when we prepare a model for prediction of drugs or efficacy. So, but the only problem is that the sequencing of these tumor genomics, it creates so much of the data that a lot of time you don't know what to use and what not to use. So the question is that, can we really draw some of the conclusion out of the raw CNV data, copy number variation data, or mutation data? And answer is yes. So two brilliant engineers from IIT Delhi, whom I worked in a company in Bangalore. And before even I present, I would like to tell you that this data belongs to the company, and it was presented in a couple of conferences. So I'm also sharing with you. It's already published also in two or three of the papers. Also presented in AACR and other conferences. Yeah, so what they did, they sort of basically simulate the signaling pathways in tumor cells based on a very similar principle, like, the Google Maps. So when you put the information that, okay, this is the destination and this is your location, and then Google Map give you the idea based on whether the road is blocked, whether the traffic is high, and everything. So similarly, they created a model which sort of take into consideration not only one mutation, but multiple mutation, multiple sort of copy number, if the copy number is high or low, and all these information. And then they prepared a working model. And what they did, they actually covered a couple of parameters, which sort of, like 5 or 6 or 7 key proteins and genes, which are responsible mainly for either proliferation or cell survival or migration of cells, like metastasis, angiogenesis, apoptosis, immune regulation, metabolism, all these things. And then using this, they prepared a working model, and the proposed simulation model has all these informations, and then whether a gene is deleted or gene is overexpressed, and accordingly you can take the call. So this company Cellworks, they have actually conducted trials on some of the AML patients as well as on solid tumors. This slide is not very clearly visible, but let me tell you that this patient, they were treated with this drug called celecoxib. And when they've got the genomic data, it was modeled using the model, they realized that there is AMPK pathway, and another one is mTOR, which was also sort of hyperactive. And another drug which model predicted which could work was metformin in a combination of celecoxib. And when they treated the patient with both the drugs, they realized that it worked. If you see that in initial days of the treatment, when the patient was treated with single drug, he has to go undergo for the blood transfusion every almost 7 or 8 days. But after treatment, this interval becomes almost 26 or 27 days. So he has to undergo blood transfusion less often, although eventually the patient died. But this is one of the working example where prediction matches with the real results. Another case study which I would like to share with you is also a patient which was given this drug called BEZ235, and another one was bortezomib. So bortezomib was a proteasome inhibitor, and the patient was on bortezomib, but then patient stopped responding. When we got the tumor genomics, we realized that this patient has a very high copy number of AKT, which leads to ultimately hyperactivation of mTOR pathway. So basically, our model that predicted that it also required mTOR inhibitor or AKT inhibitor, which should also be given in combination. And then when this mTOR inhibitor was given in combination of bortezomib, so it worked beautifully. Also, whatever we predicted, we also validated in vitro using the experiments in cell culture. And we predicted that when we given only the bortezomib alone, you could see that the cell proliferation is very high. When bortezomib was given with mTOR inhibitor at a lower concentration, the cell proliferation was predicted to be low. And then with a higher concentration, the cell proliferation was predicted to be further inhibited. And this is the real data, experimental data, and what we found was pretty much exactly the same. Not only that, we predicted that NF-kappa B could be super active or higher, like in terms of activation when there is only single inhibitor, because resistance was coming. And when the combination was given, the NF-kappa B activation was less. And similarly, cleaved PARP, if you know, it's a DNA repairing enzyme, and once it is cleaved, so it means the repairing is not happening and cell is going to die, and that's what we want. So if you could see that the cleaved PARP means cell death is higher when a combination was given, and that's what we predicted. But if you see the real data, it also matches that when like bortezomib and this BEZ, the inhibitor of mTOR, both were given, the NF-kappa B nuclear amount was lesser, cleaved PARP was higher, and this is just the loading control. So just to give you the idea that the model worked beautifully, and in most of the cases, the predictions were matching. So the question is that using this model, can we really challenge the existing clinical rules? And answer is yes. And I'll give you one example. In some of the tumors, especially if you think about, like, you know, this drug called lenalidomide. So in AML, in case of AML, there is a 5q deletion of chromosome. The Q arm of 5th chromosome is deleted, and it is considered that if there's a 5q deletion, okay, and if you give lenalidomide, right, so it should be basically a responder. But what we realized that when we give lenalidomide in this patient, it comes out to be a responder, and the reason being because a lot of genes which are present on 5q, they got deleted, but the pathways is that, like, pathways was connected in such a way that, you know, other genes which were acting as a complementary to other pathways, and those proteins were higher in this particular patient. So the idea is that if you have the whole genome information, whole mutanome information, you can predict a better outcome. That is the whole idea. So, in a way, the proposal is that before even you get on the treatment, or a clinician get on the treatment, started with standard of care drugs, it make more sense that if you have a tumor genomic information, prepare a tumor signature with key genes like maybe 200 or 150 genes, and then use the modeling, either this existing modeling, and I'm sure people with expertise like Dr. Chaitanya, who also presented on modeling. And then those models can be sort of used to prepare like a virtual patient avatar that, okay, this is the patient, these are the genes which can be targeted, these are the genes which cannot be targeted at all. And then through the process of elimination, you can sort of, select a drug uh, uh, from the existing pool. And sometimes even the repurposing also works, because a lot of time it happens that the drug is approved for, let's say, a colon cancer, but it is also working in the gastric cancer or working in the lung cancer. So it depends, as long as delivery is not an issue. So these are just the take homes, that signaling networks are complex, and mutations in tumor, it further adds to the complexity. Developing precision medicine workflow is awesome, but definitely it is good only if it is enabled by a very solid modeling and the right way of simulation of sequencing data. And then you can make predictions, any predictions, but then the predicted outcomes make sense only if they are validated on first a small set of patients, and then of course a large set of patients retrospectively as well as prospectively. So I think that was the major part of my presentation. But before I end, I would also like to talk about something which is related to, I would say, off the target talk, which is about non-conventional drug targets. And this is actually based on a lot of facts and a lot of evidences. But I would just like to give you one of the idea that when, let's say, there is a blood flow in the system, in vasculature, so there are a lot of things which also cells face mechanically. So there is a pressure, there is a shear stress, there is a stretch, also the velocity of blood. It's everything is not about the chemicals. Everything is not about the cytokine. Everything is not about the messengers. So there are physical properties also which a cell face on day to day, or maybe every time. Imagine the blood flow. So that's what is happening with endothelial cells. And then we also propose that if endothelial cells is facing that kind of stress, then probably the tumor angiogenesis is also governed by the microenvironment. Not only the chemicals, not only the pH, not only the messengers or the hormones, but also how stiff the environment is, how loose the environment is, how is the flexibility, how is the pressure, everything. And I would like to share some of the data which was published by one of the co-worker in Nature Cell Biology. And what they did, they have used these fibroblast cultures, and when they culture it on glass surface versus they culture it on a less stiffer substrate, like maybe like a rubber. So they found that their shape, their morphology, the phenotype, it completely changes. If you could see here versus here. Also, if you see that when these fibroblasts are cultured on glass surface versus on soft surface, their gaps between the cells and the way they proliferate is also very, very different. So this is something very interesting. And then I would also like to share another data with you, which is of course, which is not published by me. It was a paper published in Cell by Engler et al. And what they did, they take these stem cells and then they cultured it on elastic substrate, which were soft, a little bit stiffer, and stiffest. And then what they did, they kept all the conditions same. The cells were cultured in 10% serum, there was a collagen coating, and everything else was same, the CO2 and everything. But what they realized that the cells which were cultured on soft substrate, they eventually, after 4 days of time, they becomes nerve cells. The cells which were cultured on stiffer substrate, they become muscle cells. And the cells which were cultured on stiffest substrate, they became the bone cell. So the idea is that the microenvironment, the surface where cells, what cells are facing, that also matters. And we culture everything on non-plastic surfaces in tissue culture. So when I did this experiment using endothelial cells, what we saw that endothelial cells which are cultured on stiffer substrate, which is like 10 kilopascal versus soft substrate, they do secrete higher amount of VEGF as compared to cells which were cultured on the soft substrate. Not only that, they're signaling the molecules which get phosphorylated and related to the pro-proliferative pathways. It also varies. If you could see that soft versus stiff, so the phosphorylation of S6 is higher, which leads to protein translation and more protein synthesis. So it seems that these things also matter, and tumor vasculature is not only governed by VEGF, but there is more side to it, and which is also the physical side. Now we also try to sort of validate these results using this tube formation assay. What this assay does, that it basically align endothelial cells into a capillary like structures, and when there is a more alignment, more tubes formation, it is considered that the environment is more pro-angiogenic. So what we found that on low stiffness and on higher stiffness, you could see a clear-cut difference in the capillary like structure formation or tube formation. So it looks like the stiffer environment sort of promote tumor angiogenesis. Another data which I also would like to share, this is a traction microscopy data. Completely, I can say that you can correlate it very well with the synthetic experiments that you culture these endothelial cells and put VEGF, and when you measure those traction force, which can also lead to more migration of cells and more blood vascular formation, more vessel formation, more sprouting, what we realize that there are more hotspots, more contractile forces when cells are treated with VEGF. So these are some of the preliminary observation that at one end, we are talking about targeting of receptors, targeting of intracellular molecules to control proliferation and survival. We are talking about targeting mutations so that the treatment could be personalized. So that is one area, but another is like, this microenvironment where there are things beyond targets, beyond protein, beyond expression, which sort of leads to tumorigenesis. I'm sure they're connected, but these are some of the areas which could be explored more. So I would like to end my presentation with thanking all of them who have contributed for this work, a few of my mentors, and of course funding support from NIH and now IISER Bhopal, also the Harvard Med and Lilly for providing reagents and also funding some part of the work, and of course the CellWorks where I presented some of the modeling data, which was not done by my team, but was done through the collaboration and when I was employed at CellWorks. Yeah, thank you very much. Hello?
Hello, thank you so much, sir, for a very informative talk. We now open the floor for any question and answers from the audience.
Were slides moving, by the way, later on? I don't know if I hope that the presentation was not stuck at one point.
No, yes, sir, we could follow the slides and they were moving.
Okay.
So if there are any questions, please write them on the Q&A box.
Thank you, sir. Thank you for your talk. We have one question in the box by
Okay.
Abhinan's.
Yeah.
So
Can
at the
you?
time of
Sorry, because I could not see Q&A. I mean, I'm clicking, but it's not opening up.
okay. So the question reads: So at the time of pregnancy, when there is an angiogenesis, can tumor cells grow at that time as well?
Well, yes, if a pregnant woman can also develop a tumor, but not in the embryo. I mean, there is a regular angiogenesis and there is a pathological angiogenesis, and that is why one of the exclusion criteria of using angiogenic inhibitors for the tumor treatment is that a woman should not be pregnant. Okay, so Avastin or any other uh, angiogenic inhibitor is not given uh, to the pregnant woman, even if she has a tumor, because it can impact the regular angiogenesis.
Thank you, sir. There's one more question. Is it okay? I think this right. Yeah. So is it possible to do mathematical modeling without data? If yes, how?
Without data, no,
Yeah, that's what the question means.
no, if we are talking about modeling of signaling molecules, although I'm not an expert in mathematical modeling, but I can definitely share some of the basics. So if you're doing modeling, so modeling is done based on some information, right? You will be giving some sort of inputs or coding when you do programming, that okay, what means what. So definitely you need some data, right? You need at least a data to start with. So for example, in this model, let's assume a patient, okay, which has a gene like AKT, okay, and AKT is highly expressed. So it means the model should predict, and AKT is a pro-cancer gene. So model should predict that, okay, this cancer is like pro-growth. It is a pro, AKT is super active. It is pro-proliferative. But if you don't give this information to the model, and if you, let's say, instead of putting pro-proliferative, if you put AKT as an apoptotic gene, so it will give you a different result or different outcome. So you definitely need the information, right? You need data.
Thank you, sir. I think that wraps up the Q&A because there are no more questions in the box. All right. So I'd like you all to join me in thanking Dr. Amjad for his talk. Thank you, sir. That was a really wonderful talk.
Thanks, Prabhu.
Thank you for presenting, sir.